phases. There's going to be a cephalic phase, which maybe refers to your head. The gastric probably refers to your stomach. And then, of course, the intestinal, maybe your intestines. All right, so the cephalic phase is going to be this. It's going to involve your senses, your hearing, your seeing, your smelling. So if you hear food, see food, smell food, or think about food, that actually starts your digestive process. You might recognize that if you've ever walked by someplace and smelled something, your mouth starts to water, or makes your stomach begin to grumble. It's going, okay, yep, food, let's go. Let's start making some stuff so we can digest food. It's a rather anticipatory process. It actually goes to your hypothalamus and initiates your vagus nerve. And so get that vagus nerve down in your mind, and we're going to activate the vagus nerve with my cephalic phase, because we're going to see the vagus nerve in about 10 minutes from now. For the moment, the gastric, let's just say it's going to be the beginning of digestion, regulated by hormones. The intestinal, it's kind of hard to summarize, but it's going to be definitely the absorption of nutrients, with lots of hormone influence. You guys get more hormones. So let's start looking at this. Let's go to the cephalic phase. So you ever had your mouth water when you smelled something, saw something, or started to think about food? And we know that there's 3 major types of salivary glands in our heads, because I'm sure you all studied that over the weekend, right? The parietal, the sublingual, and the submandibulars. And in that saliva are 2 important chemical substances that we can use to begin chemical digestion. One is going to be salivary amylase, which will begin starch digestion. It likes a minorly acidic environment, 5 to 6 pH. Lingual lipase will begin lipid digestion. Again, around 5 and 6 is its happy nirvana environment condition to exist in. So now, when you see food, hear food, smell food, think about food, my brain goes, okay, there's going to be food in there. We're going to activate the vagus nerve, and specifically the parasympathetic branch of your vagus nerve. This word parasympathetic here. Come on, show up. That word right there. Now, that parasympathetic nerve is going to go to a bunch of glands that are found in your duodenum called Brunner's glands. So those parasympathetic fibers go to the Brunner's glands and increases mucus production. It's going to take you a while to make that mucus. So it's like it needs a head start. And you want to coat your duodenum with lots of good snot-like mucus. So when the food finally does get there, we're going to be protected from that pH environment. So the parasympathetic activates Brunner's glands. Brunner's glands produce mucus to coat your duodenum. Now keep in mind that the sympathetic nervous system and the parasympathetic are part of your autonomic nervous system, right? And it's a teeter-totter-like idea, sort of meaning this. If this end of my whiteboard marker is the parasympathetic and it's elevated because it's more active, that means we're going to be diminishing the activity of the sympathetic. If I increase the sympathetic, we have to turn down the parasympathetic contribution to that organ, to that structure. So what's the nickname of the parasympathetic nervous system? Rest and digest. That kind of makes sense since we're doing digestion, right? So you're going to have your rest and digest, activate the Brunner's glands. But now we have to think about the other side of the coin with this. We're going to come down here, all right, to where it says increased sympathetic activity. Now that's your fight or flight system, I should say. If the parasympathetic increases Brunner's gland activity, then the sympathetic is going to do just the opposite. It's going to decrease Brunner's gland activity, shut down mucus production. Think about the consequence of that. Once you learn that Brunner's glands produce mucus that coats the duodenum, I'm not going to have that mucus there. So when really acidic food gets there, now the epithelium is exposed to that really low pH. You might get what we call a duodenal ulcer perhaps, because there's no protection from the mucus there. This is why folks that are stressed get ulcerations a lot, because the stress tells the Brunner's glands, apparently we're in danger, let's not make mucus. You have to think about it evolutionarily. Right? Going back 50,000 years ago, danger for you was a predator, it was a fire, a volcano, or a warring tribe. Get the hell out of here, run away from danger. And you have a choice. Run from danger or digest your food. You can't do both. Take your pick. And the body says, well, I'd rather live another day, so let's sacrifice digestion and devote energy to running away from danger. Now today, you guys don't have volcanoes and warring tribes and predators as your danger, right? It's jobs and exams and relationships and nursing programs. That's your stress. It still does the same thing. Increases sympathetic activity, decreases Brunner activity, less mucus, and we have now an opportunity for maybe irritating the epithelium. And we'll kind of visit that again in a bit, probably Wednesday. But now when we're going back to the parasympathetic, we're going to be doing in a moment a whiteboard discussion. And we're going to be using acetylcholine, remember that's what ACH stands for, and things called G, parietal, and chief cells. And we're going to be looking at those tonight. Now half of you have no idea what nerve plexus is going to be, and half of you do know it, because I made a mention of it in lab tonight. You're looking at what's called the submucosal plexus. And that's the plexus that you want to write down in terms of which nerve plexus it is. The submucosal plexus, and we'll point that out to you in lab tonight for the anatomy part. All right, now this is just to get your head wrapped around the idea of what's called the gastric gland. For the moment, this is going to be the top of your stomach, and those of you who haven't had lab yet, you'll learn more about this tonight. But there's this thing called the gastric pit that leads to what's called the gastric gland. And we're going to learn that in the gastric gland are various different types of cells, such as parietal cells, chief cells, and enteroendocrine cells, all right, hormone-producing cells that make that hormone called gastrin. We're going to map out how this cell communicates to these two cells. We're going to do that on this big whiteboard here. But what I wanted to point out is that not all gastric glands have these cells together. This is just an overall general view that if you went to a gastric gland, you might find these cells in that gland. You'll learn in lab it's very regionally specific. Okay, let's spend some time talking about the hormonal signaling that takes place with those cells we just looked at. So I'm going to use this whiteboard, and you're going to, maybe if you want to participate along with it, turn your paper sideways, landscape. And then definitely when you're done, you want to photograph this to make sure that I or you didn't make a boo-boo. And we're going to start off by coming up here and doing a vagus nerve idea, coming down to right here. And then it's going to have a synapse with the little dendrites of our submucosal plexus. And this is going to be our submucosal plexus. That's this thing. Even though it looks like one neuron, that's our diagram representation of that. And we know that that nerve is going to release a neurotransmitter called ACH. Now with this, that ACH is going to go to this cell right here. And let me draw the cell and put a receptor on it. Let's put a receptor right here. That'll be an ACH receptor. The name of this cell that is in your gastric gland. So we're looking at, just to put it in context, we're looking at these little turquoise green cells right here. And there are nerve fibers coming up to them that we can't see in the diagram because nobody is going to let me actually show you guys a diagram that have it. But I know in my mind there's going to be a synapse with nerves to these cells. That's what this is. And this is going to be what's called a G cell. And when the G cell gets stimulated by acetylcholine, right, it's going to produce a hormone called gastrin. And that's going to go into circulation. It's going to travel through your entire body. It'll be in your toe, it'll be in your foot, in your nose, in your ear. But when it finally gets back to the stomach, there are going to be cells in the stomach that have receptors for that hormone. And one of the cells we're going to look at here, let me draw the cell and give you a receptor for it. So let's put that receptor right here. Give it a different shape than our acetylcholine receptor. And we'll draw that right into there. And that's a gastrin receptor for this thing called a parietal cell. Well, that seems pretty straightforward. The vagus nerve stimulates the submucosal plexus. It releases acetylcholine that lands on a G cell. This is a neural connection. In response, the G cell produces a hormone called gastrin. I know what hormones are. They are a form of intracellular signaling that affects the behavior of a cell. So we know what goes into the bloodstream. Finally gets back and activates a parietal cell. But there's more than just gastrin. So what I'm going to show you now is another acetylcholine receptor. Right? We're going to put it maybe right here just for the sake of space. We're going to show acetylcholine going to that receptor. In your mind, what I want you to realize, that we're going to totally ignore Scott, is that this ACH came from this nerve. We're not going to show nerves going to every single one of these cells. It's going to make a really crowded diagram. So just realize that this is happening right there as well. Every time we show ACH, it's this idea happening with ACH. What, Scott?
I'm just seeing which cells up there are the G cells.
Huh?
We're just wondering if any of those cells are G cells.
Yeah, the one that secretes gastrin. That secretes gastrin.
The enteroendocrine cells?
Well, yep, that's what it is. You with me so far? Okay. Now, how many of you like a little something before dinner? Y'all are lying. Okay. How many of you have a little drinky poo before dinner? Sometimes. Right? So think about this. Why culturally have we evolved to have alcohol before we have a meal? Somebody must have realized that the consumption of this substance somehow, for some reason, made eating raw meat a lot easier to do. Right? And maybe unripened fruits. Because we're talking hundreds of thousands of years ago. All right, so alcohol is one of the reasons why you guys drink before dinner. It activates G cells. It's a stimulus to increase G cell activity. All right, so
that's also why it can give you reflux then, because
we'll get to that. Hang on to that. We'll get there. Now, so Taz has had, 4 or 5 glasses of wine before dinner, 2 or 3 glasses of wine with dinner, but she can hold her own, okay? Don't be surprised by size. I have a friend, she's a cancer surviving friend of mine, and Suzanne is about this tall, and God, don't tell her I'm saying this. Probably weighs about 132 pounds, soaking wet maybe, at best. No, she's not.
Oh, she's not.
But she will outdrink all of you. I will guarantee, I will bet my life on it. I will give you all of my retirement fund, all my future salary. She will outdrink every single one of you. I've seen her consume an entire bottle of tequila and walk a straight line.
Wow.
Yeah, she's serious, okay? And we'll get to alcohol maybe consumption a bit later. But the idea is that don't let size fool you. Now, after she's had her 6, 7 glasses of wine, she's had her dinner. How many of you like a little something after dinner in the form of a dark black liquid with maybe some milk and some sugar? All right, or some kind of, you're so boring. Right? So who drinks coffee or tea after dinner? Again, why? Where'd that habit come from? because we realize that caffeine also stimulates G cell activity. We have lots of things that'll trigger G cell activity. I'm running out of shapes, so let's just make it a multi-shaped structure. And then we'll also have, we'll just do this, a little odd shaped thing. Small proteins. I can make an M, there we go. Small proteins, amino acids, they can also help stimulate G cell activity. So we got lots of things that'll activate a G cell. Now we've got apparently two things that'll activate a G cell. The hormone gastrin, the neurotransmitter called acetylcholine. There's also another one that we'll put in purple right down here. And we're going to call them H2 receptors. And they're going to be responding to histamine. So we're going to show histamine going up to that receptor. And the histamine is going to come from a cell that we're going to call ECL cells. And they are going to be activated by stretch. Or if you want, distension of the stomach wall. So think of your stomach as a big balloon. The more you blow the balloon up, the more you're stimulating the ECL cells. And the more you stimulate the ECL cells, the more histamine they release. The more histamine, which is a local signal, it's not a hormone, we're going to activate parietal cells. Well, now the only question is, what in God's name do parietal cells make? And they, quote, produce hydrochloric acid. They don't actually release hydrochloric acid, because that would kill them themselves. But that's how we talk about it. They release HCl. We'll talk about how they do that a bit later tonight. Now, I've got G cells, I've got parietal cells. We also have in your gastric pit, we're going to have a cell right up here that we're going to call chief cells. Exactly. So that's why you have to say now, chief cells of the parathyroid, chief cells of the stomach. What kind of chief cells are we talking about here? Now, chief cells are going to release something called pepsinogen. What do you guys know about pepsinogen, just based upon the name?
It's not active.
It's not active, right? Remember angiotensinogen, right? This is a pepsinogen. Ogens, no bueno. They don't work. That's an inactive form of that precursor. So I need to turn into the active enzyme called pepsin. And to do that, I need the benefit of hydrochloric acid to do this. Because I need a pH of about 1.5 to 2.0, somewhere in there. I need a really acidic environment to activate pepsinogen into pepsin. And hydrochloric acid helps to provide that really acidic environment. That's 1.5 to 2.0. Speak up so I can hear you. I can read lips only a certain extent. Now, I can see right off the bat one benefit of needing hydrochloric acid, so I can do this. Does anybody know any other benefit of hydrochloric acid in your stomach? Because which?
Kills microbes,
Kills microbes. Why do we need to do that?
so that we don't get infected by them, because they're on our food.
What, you only have a 5-second rule? I have a half-hour rule,
But
dude.
what if I had the 3-day rule? Then I'd really need to kill the microbes.
No, that's why we actually have this hydrochloric acid, more of a 3-day concept. Again, go back in time. You're walking along the plains trying to collect fruits and veggies and roots and berries. Come across this gazelle that died 2 days ago out in the nice Savannah sunshine, right? Hell yeah, as gross as that sounds, you're not going to pass it up. You're going to eat it, right? And we need that digestive ability to help kill off any pathogens that might be in that food that we're consuming. So that's another reason for needing hydrochloric acid. But also to break chemical bonds. I mean, that's where your energy comes from, the actual breaking of those covalent bonds and the energy released from those bond breaking. So, to do chemical digestion of whatever happens to be in your stomach. Now, if we look at this and go back to our ideas of regulatory pathways of hormones, I've got a runaway freight train here. Signaling lots of G cells, releasing lots of gastrin, parietal cells making lots of hydrochloric acid. How do I know I don't make too much hydrochloric acid? I need some kind of a negative feedback pathway. So we have another group of cells that are in our intestines. I'm sorry, in our stomach, pardon me, wrong part of the body just yet. Ooh, I really flatten that thing out. That still works, oh my God, okay. Blood red. These guys are called D cells. And they, in response to hydrochloric acid, are going to produce a chemical substance that is becoming a mystery to us and a wonder. We thought we knew about it, and now we're realizing we don't know very much about it at all, called somatostatin. Somatostatin is in circulation. Do it this way. And it'll do a negative feedback, I want to give the wrong impression here, on G cells. It'll do negative feedback on parietal cell activity. It'll do negative feedback on chief cells. So it helps to control all of these guys. I got a better arrow. Now there's two other stimuluses that we didn't talk about, or two stimuluses we didn't talk about for chief cells. We still have ACH going to all of our cells. Doesn't matter where you show the receptor. Maybe just keep the shape somewhat the same. So there's ACH, and I know in my mind it means this idea. Vagus nerve to submucosal plexus. We're just trying to not make a complicated diagram with this. So if you want, you could draw a little thing like this over here. Just to remind you of vagus nerve and submucosal plexus. But it also has receptors for gastrin. So we can put gastrin over here if you like. I did it with a round shape. Doesn't matter where you show the receptor. Just need to remind yourself that gastrin, having come from the G cells, goes in circulation, can also activate a chief cell. Now let's focus on this for a moment, because we're going to have to come back to this idea. If I have a G cell releasing lots of gastrin, and I start eating a lot, start stretching my stomach out, I get lots of signals, right, telling the parietal cell to make lots of hydrochloric acid. The more hydrochloric acid I create may or may not do this. What you want to think about is the lower the pH becomes. Because what we're really looking at is simply just the hydrogen ions. So the more hydrogen ions that can go to a D cell, the more somatostatin we're going to create. And the stronger the negative feedback is. So the fewer hydrogen ions, then the less stimulus, which means the less somatostatin we're going to be able to create. We're going to have to look at that a bit later tonight, maybe in lecture.
So you're saying that, okay, so let's say you wolfed out a big meal, your stomach's real distended, all this stuff going, but it'll take a while for the pH to drop.
Exactly.
It takes a while for your stomach to make all that acid. Mm-hmm. And then when it finally gets there and it's acidic enough, it shuts this machinery back down by the presence of all this hydrogen.
Yeah. So it's a nice little self-adjusting. I'm going to try and find some videos for you guys to look at. Like again, I try to follow when I have some time, these world competitive eaters. These guys are just astounding me with how much crap they can eat. And it's people that can eat 15 to 20 pounds of food in an hour at most.
That's insane.
Have a lot of stretch and a lot of this going on, and it's kind of a nice little self-regulatory system going on with this. Which makes sense because the more food I eat, right, the more I need of this stuff to go along with it. All right, so that is the hormone regulatory pathway for signaling in the stomach. Right? I am not going to ask you to reproduce this on an exam. I did that once a long time ago, and I still regret it to this day. A, in terms of grading, but also how long it took you guys to do this. But, right, you're going to want to be able to reproduce this. Because if you can reproduce it, you can answer whatever questions I can come up with. Right? So it'll be based upon the diagram, because picture's worth 1,000 words. I'd rather see this and read about it in word formation. So photograph this. Then we're going to move on to the next little idea.
So the only thing that stimulates the D cell to increase or decrease the somatostatin is the presence of hydrogen ion.
For us, yes. All right, this is actually a lot more complicated than what I gave you guys. So this is sort of like, let's call it
Close notes.
high school level biology. Then we're skipping the college level and we're skipping the graduate level. This can get really complicated. This is enough though to get through nursing school and to understand why things work the way that they do for us. But if you get into the molecular level, it's actually kind of cool, but this is as far as we need to get. All right, let's now look at how parietal cells actually produce hydrochloric acid with this. So to give you a bit of a but a refreshing moment, I'm going to give you guys a 5-minute break right now. So we can have an uninterrupted time span of looking at how this works. Take me about maybe 20 minutes to do so. And I don't want you on the verge of loss of attention span toward the end of this. So let's give you a 5-minute break now, take photographs of this, and then in 5 minutes we'll see how